Hello, my name is Louis and this is my PowerPoint on research methods, key terminology and issues. Qualitative research is a methodological position that emphasizes words rather than quantification, i.e. numbers, in collection and analysis of data. And that's from Smith, M.F. in 2010. In other words, qualitative research uses words and opinions instead of statistics and numbers, as well as also involving individuals' interpretations, meaning it is subjective. This type of research is used in the earlier phases of research projects. Furthermore, data is shown through words, pictures or objects, and the researcher may roughly know what they are looking for. Overall, they aim for a complete and detailed description of the research. A sporting example of qualitative research could be: how does an athlete's motivation help his development in sport? Quantitative research is gathering data that is quantifiable through using statistical, mathematical or computational techniques. Quantitative research points. In contrast to qualitative research, it is recommended that quantitative research is conducted in the latter parts of an investigation, and therefore the researcher knows what they are looking at. To achieve the correct results in this, all aspects of a study is designed before any data is collected by a questionnaire, for example. This type of research is objective as it seeks precise measurement and subsequently is more efficient and is able to test a hypothesis, but however it misses contextual detail. A sporting example of this could be: is there an increase in the amount of injuries in rugby games from the age of 13 to 18? Reliability. Reliability is: if you carried out a research again, you would get the same or similar results. And that's on Adams et al. 2010, page 89. In other words, it means if any investigation was completed again, would the new investigation have the same results as the previous investigation? So points linked with reliability. To make sure something is reliable, it is important it comes from a reputable source, which could be an expert in its particular field who is unbiased and use an appropriate method to collect the data. Someone who may not be reputable could be someone linked with the research or would have a reason to skew the results. An example of this could be asking a big food brand such as Pringles or Mars if their food items have decreased in size over the last 5 years. It is likely their results will show bias and unreliability due to them not wanting to provide information that is self-deprecating. A sporting example of reliability is: how does caffeine affect a 100-meter sprinter? For the results to be reliable, the results should be similar for each subject, and that could be a slight increase in time for all of them, or no increase in time for all of them. So the next point on the list is internal reliability. This links with the consistency of results within a test, including data collection, analysis and interpretation. Also, links with whether the experiment was completed in an environment where conditions can be kept the same, e.g. lab. However, this hinders the external reliability, as it doesn't actually replicate the conditions of an activity, e.g. the weather. But on the other hand, it does give a more precise measurement. Factors that affect external reliability are human error, day-to-day differences, and test length. As the longer the test goes on, the more likely the researcher will make some mistakes. It is important for a test to be reliable, as otherwise the conclusion of a research could be deemed invalid or incorrect if similar results were not obtained when repeated. This is why it is extremely important researchers use settings with a high internal reliability and then a high external reliability to prove that both settings give good evidence that the conclusion is factual. For example, if a researcher proves in a lab that 100-meter sprint time can be increased by caffeine, other researchers may argue this information due to a low external reliability, as it doesn't take into account the adrenaline and nerves of performing on a big stage. For researchers to gather good data that can prove something, they must make sure their tests are conducted in both environments, in this case lab and track, for this to be accepted. This shows how reliability can impact an investigation massively. So now moving on to validity, which is whether you are measuring what you are supposed to be measuring, and that's from Adams et al. 2010, page 89. Internal validity links to whether the results of a study are a result of the conditions placed upon it. Linking to MacLeod from 2013, he said, "The effects seen are due to the manipulation of the independent variable and not another factor." So an example of this could be increasing control variables when investigating the effect of sleep on a marathon runner's time, e.g. oxygen mask that provides same amount of oxygen each test per second. This allows the independent variable to be the only variable, which is the amount of sleep. Internal validity has a massive impact on an investigation. This is because the study is changed to ensure the results are because of a manipulation of the independent variable. Investigations with high internal validity are good for research for this exact reason. An example of this could be Wilson testing the new tennis rackets to see how tight the string should be. Different tensions of the strings in the racket can affect the control and power a player can play the tennis ball. However, in a lab, the test wouldn't cover the difference of playing the ball on clay and grass and the difference it makes. Furthermore, the weight of a ball can change slightly depending on how damp the grass is. The next point is external validity. This links to whether the results are representative of a larger population and in the real world. It refers to the extent to which results of a study can be generalized to a larger population. This could be affected by numerous things such as the weather. For example, tests done in a lab may have a low external validity due to not being in a real-world scenario, e.g. there's no rain and constant temperature is applied. An example of a high external validity is testing sleep on a marathon runner outside on an actual course. However, a low internal validity due to the amount of other variables such as the weather and surface conditions. External validity is also really important in an investigation. This is because the test directly relates to the environment the activity happens in. This is good for the investigation because it replicates the scenario the activity takes place in, giving the experiment some credibility. An example of this testing is different types of shoes on a 110-meter hurdle runner. However, the test can be invalid due to the fact the wind can change during the experiment. For example, either pushing on or holding back the runner, giving incorrect results and possibly showing one shoe is better than the other when it's not. Similarly to reliability, for a test to have high validity, investigations should take place in actual locations where the activity happens. For example, athletics tracks for a hurdler and a lab for the most valid results. The next issue in research methods is accuracy. This is how close your measurement is to the true value. Using technology increases accuracy as it takes away human error. For example, using light gates in sprints instead of a stopwatch. This is used in sport during the NFL Combine, where light gates are used to record a 40-yard dash. Research needs to be accurate for the conclusion to be considered as fact by other researchers. If it is not, other researchers may pick holes in your investigation and claim the results are false due to the lack of precise measurement. This shows how big accuracy can affect a research project. Accuracy can have a positive impact on research when it provides enough evidence that it is correct. For example, testing the amount of sugar is safe but beneficial for an Olympic 200-meter runner to have in their blood prior to a race. However, on the other hand, accuracy can have a negative impact on research, as it can ruin investigations that don't have the money for expensive equipment, or even investigations that do have the money but the technology or data does not exist. A non-sporting example of this is that we do not know if mobile phone radiation is harmful for us, and to what extent, as there is no technology or data to work this out. Precision relates to the refinement of the measuring process, and that's from Adams et al. 2010 on page 89. In simpler words, it relates to the repeatability and reliability of the investigation, or how similar the results of the investigation are. An example of this in sport could be a basketball team completing a 4-week program of strength in the lower body. At the end of the 4 weeks, you would expect all of them to have gained a similar amount of weight on their 1 rep max for deadlift, rather than some not gaining any and some gaining significantly more. Precision is important during research, as for new data to be proved true, the test must show similar results over and over again, without variation. This impacts research heavily, as if other scientists repeat your experiment, they must obtain results similar, or otherwise your test could be proved incorrect. An example of this could be testing the positive impact a vegan diet has on a heavyweight boxer. If a researcher comes out with the conclusion that a vegan diet is best for a heavyweight boxer, but another researcher has more precise data on the comparison on non-vegan and vegan diets on heavyweight boxers, your information will be disregarded and called false. The negatives of precision on research are that it requires a high level and expensive equipment to be able to maintain variables that could affect investigations, such as oxygen levels in an athlete. On the PowerPoint, you can see 4 archery targets. This gives an idea about accuracy and precision and how they go hand in hand. In the bottom left hand corner, there's a low precision and low accuracy, showing that the black marks are very far away from the middle of the target. However, as precision and accuracy increase to the top right hand target, they narrow in and center on the center mark. This is similar to research. So the next 2 things I'll be talking about is primary data and secondary data. Primary data is when the researcher himself or herself collects the data and it is used to investigate a research problem. There are multiple ways to find out primary data, including questionnaires, interviews, and observations. An example of when this is used in sport is a physiotherapist interviewing players after the match in football to find out if they have any knocks or bruises, so it can be logged and treated right away. Secondary data is when a researcher finds previously published data. It can be used to support and boost your own findings, or even counter-argue your own data. You can find secondary data in books, journals, government publications, or websites on the internet. An example of when secondary data could be used is when professionals, such as nutritionists, use information to support their own findings, e.g., protein is excellent for repairing muscle cells. So now I'll be covering the different types of data. Discrete data consists of categories that are given names or labels. It is something that can only be counted, and each value is separate and distinct. It also has no order. Data can be recorded anonymously by replacing their category title by a numerical figure, i.e., giving football players data in the number 1 category and rugby players in the number 2 category. The numbers have no meaning whatsoever and just separate the categories. Nominal data. This is when participants are separated into categories which then can be counted, but however it does not mean one category is better than the other. To analyze the data, the percentage of each category can be calculated, or the results can be shown in a pie chart. For example, this could be grouping football players by position in the team to show how many players they have at each position. Ordinal data is data that provides a ranking or numbering system, but however it does not show the difference between each stage. This type of data is used often when asking for opinions or attitudes. For example, who are your top 5 favourite footballers of all time? Furthermore, this type of data can be used for rating scales. For example, strongly agree or to strongly disagree. Continuous data is data that can have any numerical value with an unlimited number of decimal places. An example of when continuous data is used could be 400-meter race times in the Olympics, as any value could occur. Interval data. This data consists of equal gaps between each point on a scale, but however there is no true starting point. An example of this could be the Fahrenheit scale, as the gap between each measurement is the same. Ratio data, which is very similar to interval data, consists of equal gaps between each point on a scale, but however there is a true starting point, which is 0. This means there can be no negative scores. An example to show you this could be that a basketball team is beating another team 12-4. The winning team are ahead by 3 times the amount of points. The next part of the PowerPoint I'll be talking about is interviews. Interviews are normally conducted in a one-to-one situation. They are good to get people to express views in their own perspective and not to be influenced by others. Other positives may include that unexpected data may come out and they can be used to refine findings found out through research. However, interviews are a long and difficult process to undertake properly, and the sample size of people is small. Lastly, participants can take the interview in the wrong direction, meaning relevant data is not collected. There are 3 types of interviews which are all good for different situations. Structured interviews have no flexibility as all questions are set prior to the interview. An example of this could be a post-match interview in football where the reporter is given specific questions to ask. A positive of this is that it is a fairly reliable way of information gathering, as you won't stray off topic. However, the data may be limited and of poor quality. Semi-structured interviews include questions that are prepared prior to the interview, but they are flexible. This means the interviewer could ask the person to expand on a question, which creates an opportunity to create further discussion. This is a positive as it allows the interviewer to ask for in-depth answers of a higher standard. However, as each interview is different, it will be hard to compare them to others, as each participant would talk about something different each time. An example of a semi-structured interview is a job interview. Lastly, unstructured interviews consist of a theme and with little or no set questions. Normally, this type of interview is more of a conversation, and it allows the person to expand further on answers, which is a positive. However, the interviewer may stray from his area he is questioned about and may collect irrelevant data. An example of an unstructured interview could be parts of a job interview, depending on what type of questions they ask. A good example of an unstructured question is, what is your greatest strength? Focus groups are a classification of interview but involve 6 to 12 people rather than just the one. Also, the interviewer is not too involved in the conversation and acts as a facilitator by giving a group a question so they can discuss it between themselves. The positives of focus groups include deeper discussions, and people are more comfortable in a group setting as it seems more like a conversation rather than an interview. However, similarly to interviews, the process is still very long and the sample size is not much bigger. Additionally to this, people may not equally contribute in a focus group, which can affect results. An example when focus groups could be used is during a market research, when companies, for example Microsoft, host focus groups to gather feedback about products to improve them. The next slide is about observation. When observing, the researcher is actively involved and watches to gather information. This means the data collected is subjective. Observing could be good for numerous different subject areas such as psychology and evaluation of coaching. Observation can be split into 2 different sections: participant observation and non-participant observation. Participant observation is where a researcher is actively involved and is observing from the inside. Data is recorded as field notes and it consists of own feelings and thoughts, etc. This is best used when researching sensitive subjects and behaviours. An example of this could be a psychologist asking questions and watching the behaviours of their client during an examination. On the other hand, non-participant observation is when a researcher observes from the outside and has no interaction with the individuals or activity whatsoever. An example of this could be scouting, when recruiters go to games and watch players without them knowing they are there sometimes. The positives of the observation are that it is here and now data and it is based on recall. Furthermore, the research takes place in a natural setting and it allows the researcher to pick up on behaviours that are not apparent to the participant. However, the negatives include that a researcher may not know what they are seeing, and it may be difficult to identify and record data. The biggest negative is the Hawthorne effect, which means that people act differently when they know they are being observed, but it can mess up their data. Observation, focus groups, and interviews are all part of qualitative research. Moving on to quantitative research, beginning with questionnaires. Questionnaires are a very common method of data collection. It consists of asking questions for a pencil and paper survey which the participant responds to. Information is collected through the answers of the questions rather than observing their behaviour. Questionnaires can be used for a variety of different things, from physical activity readiness questionnaires to preferences of food. The positives of using questionnaires include it is a quick and easy process. Clients can choose to remain anonymous, which is important for more sensitive topics where respondents don't wish others to see the information. It is relatively cheap, relatively accurate, and it is easy to analyse the data because the data is simple. However, on the other hand, there are negatives associated with questionnaires. These include people may not answer the questionnaire honestly, as people may see it as a waste of time and wish to write anything down on the paper. It is difficult to follow up anonymous questions, and social bias may affect the answer. In questionnaires, you can use 2 types of questions: open questions or closed questions. Open questions ask the participant to write more detailed sentences about the topic at hand, An example of this could be, what is your opinion on the amount of money footballers are paid? This question makes the respondent write more for it to be answered properly. This is effective to find out more detailed information in topic areas, but however, it makes the process longer, and as the data is not as simple as it could be, meaning it will take longer to analyse. On the other hand, closed questions ask for a short answer, which usually could be yes or no. An example of this could be, are you a professional athlete? This type of question is quick and it is easily analysed, but however, does not give us the contextual detail. For example, this could be what sport the participant plays and how much money they earn. The penultimate slide is field-based data collection, which consists of data collection in an environment which sport is played, for example a football pitch. The positives of this type of data collection are a high ecological validity, which relates straight to the demands of the activity. In football, the weather is involved, which affects the game in numerous ways. For example, speed of a ball across the grass. It's also cheaper, as it's cheap to find a playing field to conduct testing compared to a high-level lab and expensive equipment, and it is also accessible, as there are plenty of playing fields or grass spaces for data collection to take place. However, on the other hand, there are a few negatives. There is a low internal validity. Having a high ecological validity sacrifices the internal validity of the research and may mean other variables could impact data. For example, if you are testing your goalkeeper's goal kick distance, the wind could change each go and affect the data by carrying it forward or slowing the ball down. Also, it is difficult to control variables and repeat test. As mentioned before, in football the variables can change, for example wind, meaning the test is hard to repeat while also obtaining similar results. The last part of my presentation today is lab-based data collection, which is collecting data in an environment where variables are controlled, which ensure you are only measuring the variable in question. An example of this is measuring the VO2 max of an Olympic cyclist in a lab. The positives of lab-based data collection include easy to control variables, it has a high internal validity, and the test is repeatable due to the ability to control the variables such as the temperature in the room. However, on the other hand, the negatives are a low ecological validity, as the test doesn't include other variables such as the weather, and it involves having technical and expensive equipment which some researchers may not have.